These 5 learning objectives outline the essential genetics competencies for your certification exam. You'll master inheritance risk calculations, recognize key dysmorphic features, understand chromosomal processes, interpret genetic test results, and apply counseling principles in neonatal care. Each objective directly correlates with high-yield exam content and clinical scenarios you'll encounter in practice. These skills form the foundation for genetic assessment and family support in the neonatal intensive care unit. DNA serves as the molecular basis of heredity within every cell nucleus. Picture an instruction manual written with just 4 letters, A, T, G, and C, representing adenine, thymine, guanine, and cytosine. These nucleotides follow strict pairing rules, A with T, G with C. This complementary base pairing explains how DNA replicates accurately and why certain mutations occur when these rules are disrupted. The remarkable fact that humans share 99% identical DNA provides crucial context for genetic counseling. That remaining 1% accounts for all human genetic variation. When families struggle to understand how their baby developed a genetic condition with no family history, this concept helps explain de novo mutations, those arising spontaneously rather than being inherited. Genes represent functional hereditary units, specific DNA segments that code for cellular products. The human genome contains approximately 20,000 to 25,000 genes, each occupying a specific chromosomal location called a locus. This precise mapping allows us to pinpoint where deletions, duplications, or mutations occur when we receive genetic test results. The classical teaching that one gene equals one protein no longer holds true. Through processes like alternative splicing and post-translational modifications, a single gene can produce multiple protein variants. Consider the CFTR gene in cystic fibrosis. Different mutations affect different protein domains, explaining why some infants present with severe pulmonary disease, while others show primarily pancreatic insufficiency. This molecular variability translates directly to bedside care. When we encounter an infant with multiple congenital anomalies, recognizing patterns becomes essential. The same gene mutation can manifest differently based on which protein function is disrupted, genetic modifiers, and environmental factors. This complexity underscores why genetic counseling requires both molecular knowledge and clinical correlation. We're not just interpreting lab results, we're helping families understand their baby's unique presentation and prognosis. Genotype and phenotype represent 2 sides of the same genetic coin, concepts that shape every genetic consultation in the NICU. The genotype encompasses an individual's actual genetic makeup, the precise DNA sequence inherited from both parents. We cannot determine genotype through physical examination alone. It requires genetic testing, whether karyotype, microarray, or molecular analysis. Take the notation 47XX plus 21. This genotype tells us the infant has 47 chromosomes total, with 2 X chromosomes indicating female sex and an extra chromosome 21 causing Down syndrome. The phenotype consists of all observable characteristics and clinical traits, essentially everything we can detect through physical examination, imaging, and clinical assessment. When we examine an infant and note hypotonia, upslanting palpebral fissures, and identify a cardiac defect on echocardiogram, we're describing the phenotype. These observable features result from how the underlying genotype interacts with developmental processes and environmental factors. The relationship between genotype and phenotype proves more complex than simple one-to-one correlation. Multiple infants with identical 47XX plus 21 genotypes will share certain features. The classic facial characteristics, increased risk for atrioventricular canal defects, and developmental delays. Yet each infant presents uniquely. One might have severe cardiac disease requiring immediate intervention, while another has only a small VSD. This phenotypic variability occurs because genes don't act in isolation. They interact with other genes, epigenetic factors, and the prenatal environment. In practical terms, phenotype guides our initial clinical suspicion, while genotype provides definitive diagnosis. When admitting an infant with hypotonia, single palmar creases, and protruding tongue, we recognize the phenotypic patterns suggesting Down syndrome. However, confirming the diagnosis requires genetic testing to identify the specific genotype, whether standard trisomy 21, mosaic form, or translocation. This distinction matters for recurrence risk counseling and identifying which parent might carry a balanced translocation. Remember, phenotype equals what you observe at the bedside, genotype equals what the laboratory reports. Both pieces complete the diagnostic puzzle. Chromosomes serve as the packaging system for our genetic material, thread-like structures that organize and protect DNA during cell division. Picture taking a 2-meter strand of DNA and wrapping it around protein spools called histones, then coiling and supercoiling until it condenses into a structure visible under a microscope. This remarkable compression allows each cell to contain all genetic information while still fitting within a nucleus measuring just micrometers across. The normal human karyotype contains 46 chromosomes arranged in 23 pairs. 22 pairs are autosomes, numbered from largest to smallest. Chromosome 1 being the largest, containing about 249 million base pairs, down to chromosome 22 with approximately 51 million base pairs. The numbering system helps us communicate precisely about genetic findings. When we report that an infant has a deletion on chromosome 15, every geneticist worldwide knows exactly which chromosome we mean. The 23rd pair consists of sex chromosomes, XX for typical female development, XY for typical male. These chromosomes do more than determine sex. They carry genes affecting growth, metabolism, and neurodevelopment. This explains why sex chromosome abnormalities like Turner syndrome, 45X, or Klinefelter syndrome, 47XXY, present with features beyond reproductive differences. A karyotype represents the standard visual arrangement of all chromosome pairs, captured when cells are arrested during metaphase of cell division, the point when chromosomes are most condensed and visible. Laboratory technicians photograph the chromosomes, then arrange them in numerical order, creating the familiar karyotype image we review in genetic reports. This systematic arrangement allows us to identify numerical abnormalities, like trisomy 21, structural changes like deletions or translocations, and even subtle rearrangements that might escape detection through other methods. When we order a karyotype for an infant with multiple congenital anomalies, we're looking for these visible chromosomal changes. The resolution limits mean we can typically detect abnormalities involving segments larger than 5 to 10 million base pairs. Anything smaller requires more sensitive techniques like microarray or molecular testing. Chromosome anatomy follows a standardized nomenclature system that allows precise communication about genetic findings worldwide. Each chromosome resembles the letter X or Y when condensed during cell division, with distinct structural components we must recognize to interpret genetic reports accurately. The short arm of each chromosome is designated P from the French word petit, meaning small. The long arm is labeled Q, simply because it follows P in the alphabet. This naming convention arose in the 1960s when French cytogeneticists first described chromosome structure. The consistency matters tremendously. When any laboratory reports a deletion on 5p, every clinician knows they're referring to the short arm of chromosome 5. The centromere serves as the chromosome's connection point, where sister chromatids attach during cell division, and where spindle fibers grip to pull chromosomes apart. Centromere position varies among chromosomes, creating their characteristic shapes. Some chromosomes, like 13, 14, and 15, have centromeres very close to one end, acrocentric, while others, like chromosome 1, have more centrally located centromeres, metacentric. The clinical significance becomes clear when we receive genetic reports. Take the notation 22q11.2. This precisely identifies chromosome 22, long arm, region 11, band 2. Each chromosome arm is divided into regions, then bands, then subbands, creating an addressing system as specific as GPS coordinates. When a report states an infant has a deletion at 22q11.2, we immediately know this refers to DiGeorge syndrome. This anatomical precision transforms vague clinical suspicions into actionable diagnoses. An infant presenting with tetralogy of Fallot, feeding difficulties, and subtle facial features prompts us to look specifically at 22q11.2. The standardized nomenclature ensures that whether the genetic testing occurs in our hospital or across the globe, the results speak the same language. Understanding chromosome anatomy isn't just academic. It's the foundation for interpreting every genetic report that crosses our desk and translating those findings into meaningful prognostic information for families. Homozygous means that both alleles for a particular gene are the same, either two dominant alleles or two recessive alleles. Genes come in pairs, one from each parent, and alleles are different versions of a gene. If both copies are dominant, for example, AA, the dominant trait is expressed. If both are recessive, like AA, then the recessive trait appears. In genetic conditions, being homozygous for a recessive allele is often what leads to disease. For example, a child with two copies of the faulty CFTR gene, AA, will have cystic fibrosis. Heterozygous refers to having two different alleles for the same gene, one dominant and one recessive, written as AA. The dominant allele typically determines the physical trait or phenotype, while the recessive allele remains silent. This means the person may carry a gene for a genetic condition without showing symptoms. They're a carrier. A classic example is sickle cell trait, where a person with one normal hemoglobin allele and one sickle cell allele doesn't have the disease but can pass the gene to their children. In Mendelian inheritance, traits are passed down through genes from both parents. Each person has two copies of every gene, one from the mother and one from the father. These copies are called alleles. If the alleles are different, the dominant one usually determines how the trait appears, while the recessive one may be hidden. These inheritance patterns, dominant or recessive, help us understand how traits and genetic conditions are passed through families. We'll explore specific examples later, but this framework forms the basis for understanding genetic risk. Dominant and recessive refer to how alleles influence traits. A dominant allele only needs one copy to be expressed. So if someone inherits it from just one parent, the trait or condition appears. In contrast, a recessive trait only shows when both alleles are recessive, meaning the person inherited one from each parent. A heterozygous individual, someone with one dominant and one recessive allele, will usually show the dominant trait, but can silently carry the recessive gene. Understanding this is essential for predicting who might show a trait, who might carry it, and how it can pass through generations. Chromosome numbers follow precise patterns that explain both normal development and the chromosomal abnormalities we encounter in the NICU. Every somatic cell in the human body, from neurons to hepatocytes to the lymphocytes we send for karyotyping, contains 46 chromosomes. We call this the diploid number, written as 2n equals 46, because these cells carry two complete sets of genetic information, one set inherited from mother, one from father. Think of it like having two copies of an instruction manual. If one copy has a smudged page, you can refer to the other copy. This redundancy protects against many genetic errors, which explains why most single-gene mutations require defects in both copies to cause disease. The principle behind recessive inheritance patterns. Gametes, sperm and egg cells, contain exactly half this genetic material. Through meiosis, these specialized cells end up with just 23 chromosomes, one from each pair. We term this the haploid number, n equals 23. This reduction serves a critical purpose. When sperm meets egg during fertilization, the chromosome number returns to 46, maintaining species stability across generations. The mathematics seems simple. 23 plus 23 equals 46. Yet this process represents one of biology's most vulnerable moments. Errors during meiosis lead to gametes with too many or too few chromosomes. When a sperm carrying 24 chromosomes fertilizes a normal egg with 23, the resulting zygote has 47 chromosomes, the basis for trisomy conditions like Down syndrome, Edwards syndrome, and Patau syndrome that we diagnose regularly. This fundamental concept explains why maternal age increases trisomy risk. As oocytes age, the cellular machinery maintaining proper chromosome separation during meiosis becomes less reliable. The spindle apparatus might fail to pull chromosomes apart correctly, leading to nondisjunction. Both copies of chromosome 21 ending up in one egg cell instead of separating. Understanding normal chromosome numbers also clarifies why certain abnormalities are incompatible with life. While trisomy 21 allows survival, trisomy 1 would add an enormous chromosome packed with essential genes. Too much genetic imbalance for embryonic development. This principle helps families understand why some pregnancies with chromosomal abnormalities end in miscarriage, while others, like their baby with trisomy 21, survive to the NICU and beyond. A mutation is a permanent change in the DNA sequence. These changes can occur in a single gene or larger parts of a chromosome. Mutations come in several types, including point mutations, where just one DNA base is altered, deletions or insertions, where small pieces of DNA are lost or added, and duplications, where sections of DNA are copied. Mutations can happen spontaneously or be inherited from parents. Their effects vary widely. Some cause no change at all, while others can lead to serious genetic disorders. Understanding mutations is fundamental in genetics, especially when studying neonatal conditions. Aneuploidy represents any chromosome count that isn't a multiple of 23, the most common chromosomal abnormality we encounter in the NICU. Rather than the normal 46 chromosomes, aneuploid cells might have 45, 47, or other non-multiple counts. The mechanism behind most aneuploidy involves nondisjunction, where chromosomes fail to separate properly during meiosis. Picture the cell division process where chromosome pairs should split evenly, one copy going to each daughter cell. When the cellular machinery fails, both copies travel together, creating one cell with an extra chromosome and another missing that chromosome entirely. This explains why we see both trisomies and monosomies, though their survival rates differ dramatically. Trisomy 21, resulting in 47 chromosomes total, represents our most common viable autosomal trisomy. These infants arrive in our NICU with characteristic features, often requiring cardiac evaluation and feeding support. Turner syndrome, with 45 chromosomes due to a missing X chromosome, presents differently. Lymphedema, webbed neck, and cardiac anomalies like coarctation. The key insight here: losing genetic material typically causes more severe consequences than gaining it, which explains why autosomal monosomies are virtually never viable, while several trisomies allow survival. Triploidy takes chromosomal imbalance to an extreme. 69 chromosomes, representing 3 complete sets instead of 2. This can occur through various mechanisms: fertilization of an egg by 2 sperm, fertilization of an abnormal diploid egg by normal sperm, or fertilization of a normal egg by abnormal diploid sperm. The massive genetic imbalance proves incompatible with long-term survival. When we encounter a severely growth-restricted infant with multiple anomalies who dies shortly after birth, triploidy often explains the constellation of findings. The statistic that triploidy accounts for 15% of chromosomally abnormal pregnancy losses highlights its frequency despite its lethality. Most triploid conceptions miscarry in the first trimester. The rare cases reaching our NICU present with severe symmetric growth restriction, syndactyly, and characteristic facial features, including low-set ears and micrognathia. These infants rarely survive beyond the immediate newborn period, making compassionate care planning essential from admission. This knowledge shapes our counseling approach. When parents ask why their baby has a chromosomal abnormality, we can explain nondisjunction as a random error in cell division, not something they caused or could have prevented. The distinction between aneuploidy types helps set realistic expectations and guide care decisions. Chromosomal deletions occur when a segment of DNA breaks off and is lost during cell division or DNA repair. This creates a partial monosomy. The cell now has only one copy of all genes within that deleted region instead of the normal 2 copies. The size and location of the deletion determine its clinical impact, ranging from minor effects to severe multi-system disorders we manage in the NICU. The mechanism seems straightforward, but the consequences cascade through development. When genes are deleted, the proteins they encode either decrease by half, haploinsufficiency, or disappear entirely if the remaining copy cannot compensate. Some genes tolerate this reduction, others do not. Genes that require precise dosing for normal development, particularly those involved in early embryogenesis, cardiac development, or brain formation, cause significant problems when deleted. Common deletion syndromes include DiGeorge syndrome from 22q11.2 deletion and Cre-de-Chat syndrome from 5p deletion. Each presents with characteristic features that help us recognize them clinically. The distinctive high-pitched cry in Cre-de-Chat results from laryngeal abnormalities caused by the specific genes lost in that deletion. These recognizable patterns allow us to anticipate needed evaluations and interventions from the moment of admission. The principle that deletions often cause more severe effects than duplications reflects basic genetic balance. Having 3 copies of genes means 150% of normal gene product. Potentially problematic, but often tolerable. Having 1 copy means only 50% of normal gene product. And many developmental processes cannot function properly with half the required proteins. This explains why partial monosomies typically cause more severe phenotypes than partial trisomies of the same chromosomal region. Size matters, but so does content. A small deletion in a gene-rich region might cause more severe effects than a larger deletion in a gene-poor area. Some chromosomal regions contain clusters of developmentally critical genes, while others harbor mostly repetitive DNA with few coding sequences. This variability explains why 2 infants with similar-sized deletions on different chromosomes can have vastly different outcomes. One might have isolated learning differences, while another faces life-threatening congenital anomalies. Chromosomal duplications represent the flip side of deletions. Instead of losing genetic material, cells gain extra copies of chromosomal segments. This creates a partial trisomy for all genes within the duplicated region. Where a deletion leaves cells with 1 copy of affected genes, duplication provides 3 copies, disrupting the carefully balanced gene dosage required for normal development. Gene dosage imbalance drives the clinical consequences we observe. Cells expect precisely 2 copies of most genes, 1 from each parent. This balance regulates protein production, like a thermostat maintains temperature. When duplication adds a 3rd copy, protein production increases by 50%. Many cellular processes can adapt to this excess better than they handle the deficiency caused by deletions, which explains the generally milder phenotypes we see with duplications. Consider the pathophysiology. Excess protein might slow a developmental pathway or create mild dysfunction, while complete absence of that protein could halt the process entirely. This principle holds across most duplications, though exceptions exist when the duplicated region contains dosage-sensitive genes that cannot tolerate any imbalance. Charcot-Marie Tooth disease type 1A illustrates how even small duplications cause clinical disease. A tiny 1.4 megabase duplication on chromosome 17p12 creates an extra copy of the PMP22 gene. This gene encodes peripheral myelin protein 22, crucial for maintaining the myelin sheath around peripheral nerves. The 50% increase in PMP22 protein disrupts myelin stability, leading to progressive peripheral neuropathy. While infants rarely show symptoms at birth, knowing about familial CMT1A helps us recognize early signs, like hypotonia or delayed motor milestones, that might otherwise be dismissed. The relative mildness of duplications compared to deletions shapes our prognostic discussions with families. When prenatal testing reveals a duplication, we can often provide more optimistic counseling than for similar-sized deletions. However, less severe doesn't mean benign. Duplications still cause intellectual disability, growth abnormalities, and congenital anomalies, just typically with better overall outcomes than their deletion counterparts. This knowledge proves practical during delivery room resuscitation and initial NICU management. While an infant with a known deletion might require immediate cardiac evaluation and metabolic screening, one with a duplication might present with subtler findings, requiring careful observation to identify affected systems. Nondisjunction represents one of the most critical mechanisms underlying chromosomal abnormalities we encounter in the NICU. This process occurs when chromosome pairs fail to separate properly during meiosis, the specialized cell division that creates gametes. Picture the normal process where homologous chromosomes line up and then separate, with one copy going to each daughter cell. In nondisjunction, both chromosomes travel together to one cell, leaving the other cell without any copy. This failure creates gametes with abnormal chromosome numbers. One gamete receives both chromosomes from a pair, 24 total, while the other receives neither, 22 total. When fertilization occurs, these abnormal gametes produce embryos with chromosome imbalances. If a gamete with 24 chromosomes meets a normal gamete with 23, the result is trisomy, 3 copies of that chromosome, totaling 47. Conversely, a gamete missing a chromosome creates monosomy, just 1 copy, totaling 45. The clinical consequences depend entirely on which chromosome experiences nondisjunction. Trisomy 21 creates Down syndrome, with its characteristic features and medical complications. Monosomy X results in Turner syndrome. Most other autosomal monosomies prove lethal, explaining why we rarely see them in live births. The statistic that 90% of Down syndrome cases result from maternal nondisjunction carries profound implications for genetic counseling. This predominance relates to the unique timeline of female meiosis. Oocytes begin meiosis during fetal development but arrest in prophase 1 until ovulation, potentially decades later. This prolonged arrest allows accumulation of age-related damage to the spindle apparatus and cohesion proteins that hold chromosomes together. The correlation between maternal age and nondisjunction risk provides the scientific basis for age-based prenatal screening recommendations. Recognizing nondisjunction as the primary mechanism behind numerical chromosomal abnormalities helps us counsel families appropriately. Parents often blame themselves, wondering what they did wrong. We can explain that nondisjunction occurs randomly during gamete formation, a biological error in cell division machinery, not a result of parental actions or environmental exposures. This understanding forms the foundation for discussing specific syndromes and their management strategies in subsequent discussions. Translocation occurs when a chromosomal segment breaks off and reattaches to a different chromosome, essentially genetic material moving to the wrong address. This rearrangement fundamentally differs from deletions or duplications because the DNA doesn't disappear or multiply. It simply relocates. The clinical impact depends entirely on whether genetic material is gained or lost in the process. Balanced translocations maintain the complete genetic complement. All genes remain present, just rearranged. Picture cutting pages from 2 different instruction manuals and swapping them. As long as all pages remain available, the instructions stay complete, even if they're bound in different volumes. Parents carrying balanced translocations typically appear phenotypically normal because their cells contain all necessary genetic information. However, these rearrangements create significant risks during meiosis. When balanced translocation carriers produce gametes, their rearranged chromosomes cannot pair normally during meiosis. Some gametes receive the correct amount of genetic material, while others end up with duplications or deletions. This explains why phenotypically normal parents with balanced translocations face increased risks for miscarriages and offspring with congenital anomalies. Unbalanced translocations result when offspring inherit an abnormal chromosome combination. creating partial trisomies or partial monosomies. The infant now has 3 copies of some genes and only 1 copy of others. The severity depends on which chromosomal regions are involved and the size of the imbalanced segments. Larger imbalances typically cause more severe phenotypes, often incompatible with life. The observation that 3 to 5% of Down syndrome cases result from translocation rather than simple nondisjunction carries crucial implications. Unlike standard trisomy 21, translocation Down syndrome can run in families. When we diagnose Down syndrome in a newborn, determining whether it results from translocation versus nondisjunction becomes essential for accurate recurrence risk counseling. If one parent carries a balanced translocation involving chromosome 21, future pregnancies face significantly higher risks than the age-related risks associated with nondisjunction. This distinction drives our recommendation for parental karyotyping when translocation Down syndrome is identified. Finding a balanced translocation in a parent changes genetic counseling dramatically, from discussing age-related risks to explaining specific translocation-dependent probabilities. Some translocations carry risks approaching 100% for abnormal outcomes, while others pose more modest increases. Understanding translocation mechanisms helps us provide families with accurate information for informed reproductive decisions.